Hi there, welcome to Tercen. Today we're going to talk about how to integrate cytometry data with additional information to transform it and gain new insights. Cytometry measurements alone don't always tell the picture of a patient's background. Often there are other factors that are needed to provide important context to the measurements so that meaning can be derived from them. These factors could be measurements from other analysis platforms, or they might be annotations recording important information about the sample conditions or the patient's clinical background. Integrative disciplines, such as translational medicine research, look to synthesize data from different sources to find answers to research questions. Cytometry data is often a part of this translational mix, and in this video we will show you how Tercen can combine separate data sources into visual projections. To demonstrate this, we've taken some research from a clinical trial conducted by the BM Foresight project. The trial looked at using vitamin D in treatments of allergies to tree pollen. The researchers performed three types of measurements on PBMC samples taken from the patients: a cytometry fax, a cytokine immune response test, and a cell proliferation study. Let's take a look at their data to get familiar with it. These measurements represent intracellular cytokines. Note the values are in picogram per milliliter. This proliferation data gives a ratio measure of the cell growth. See that our files have a platform type field to ID the machine which took the measurement. We'll be using that later in our projections. The fax data has marker combinations scored as percentages of cell populations, and there is a fluorescent intensities for some markers. The researchers have additional clinical data on their patients in this annotation file. They've recorded important information such as if the patient is allergic or healthy and whether they received the treatment or the placebo. As you can imagine, this kind of information is not output by a flow cytometer, but is extremely relevant to how we interpret that machine's results. Note that every patient has a patient ID. We're going to use this later. In the measurement files, a sample ID is given to each observation. It's a code that combines the patient ID, the time point of the test, and the stimulation used in the sample. This annotation file breaks out the sample ID into these constituent parts. This means we can use these individual factors for analysis, which we wouldn't be able to do with the original sample ID. So, let's create a workflow to translate our raw data with some annotations. In Tercen, a workflow is started by adding a table. But this time I'm going to do it slightly differently and select all three of the measurement tables to be imported together. Doing it this way will make Tercen build them into a single dataset. This saves you having to do it in advance on something like Excel. Columns with the same name, like sample ID, will be built together and the unique columns will be set out separately. I'll apply a gather to transform our amalgamated data into long format. This makes it easier for operators to run calculations on it. Now we can bring in our annotation tables in the traditional way. We're going to connect them, and if you're familiar with SQL, you'll recognize joins in Tercen. The important thing about joins is that there must be a field in both tables where there'll be matching information. This is called a key. Sample ID is shared by the measurement files and the sample annotations, so that is our key. The patient annotations don't have the sample ID, so we can't link them to the measurement files as they don't share a key. But they do share a key with the sample annotations, the patient ID. We can join the patient annotations to the sample annotations, and because we've already joined the sample annotations to the measurements, we have a link from the patient back to the measurement. To save some time, I built out some projections of this data so we can take a look at how these annotations can provide advanced meaning to the measurements. Let's start with a simple heat map projection. I put all of the measurements from our amalgamated files into a list on the y-axis and started using the annotations to split them into groupings. Along the top on the x-axis, we've divided our patients into allergic or healthy and whether they received a treatment or not. Down the side, we've grouped by the platform that provided the measurement. If we zoom in, we can see the timestamp where the sample was tested and the sample ID that provided the measurement. Straight away we're getting information visually that's difficult to spot in the spreadsheet. It's obvious now that there's no fax data for the stimulated samples. Only the unstimulated samples were put through the flow cytometer. Now that we have our data laid out in a meaningful way, we can apply techniques to analyse it even further. For example, we may decide to concentrate our analysis where we have a full dataset, and we want to exclude the records where we only have partial data. To do this, I applied a filter using one of the sample annotations. By dragging the stimulation factor over to filters, I can set it to include only the stimulated samples. Now that I've filtered our data, I can start looking for patterns in the heat map. There doesn't seem to be an obvious pattern here, but as we mentioned earlier, the platforms are not measuring the same thing. The cytometry data is in percentages and the cytokines in picograms per milliliter. Scaling is a technique which allows us to compare disparate pieces of information to each other by their levels of variance rather than their absolute values. By applying a scaling operator, we can move away from the measurement values and start looking at their level of variance instead. This heat map shows the data after it's been scaled. Again, nothing visible, but hierarchical clustering could put these values into logical order. In this clustered heat map, I think we can finally see a pattern in some of the markers. And by selecting them and applying another filter, we can exclude the noise and focus on the most interesting measurements. As you can see, by joining the annotations and sorting by these annotations, data can be enriched with meaning in ways that are not possible by just looking at raw output from an analysis platform. More analysis can be done as you need it, and at Tercen we would always encourage you to play and experiment with your data. So that is the end of our tutorial on analysing high-dimensional flow cytometry. I hope you enjoyed it, and I hope we've shown you some useful techniques to help with your data analysis journey. If you have any questions or comments, please don't hesitate to get in touch by email to support@tercen.com. Thanks for watching.